Skeletal muscle fibers. Each skeletal muscle fiber is a single, long, cylindrical cell containing several nuclei, which are located around its periphery near the plasma membrane. A single fiber can extend from one end of the muscle to the other. In most cases, the fibers range from approximately 1 millimeter to about 4 centimeters in length and from 1 micrometer to 100 micrometers in diameter. Large muscles contain a large percentage of large diameter fibers, whereas small, delicate muscles contain a large percentage of small diameter fibers. However, any given muscle contains a mixture of small and large diameter fibers, as seen in the longitudinal section. Alternating light and dark bands give the muscle fiber a striated or striped appearance. Figure 9.2. Origin of muscle fibers. Muscle fibers develop from less mature multinucleated cells called myoblasts. The multiple nuclei result from the fusion of myoblast precursor cells, not from the division of nuclei within myoblasts. Myoblasts are converted to muscle fibers to contractile proteins accumulate within their cytoplasm. Shortly after the myoblasts form, nerves grow into the area and innervate the developing muscle fibers. The number of skeletal muscle fibers remain relatively constant after birth. Enlargement or hypertrophy of muscles after birth in children and adults result from an increase in the size of each muscle fiber, not from the substantial increase in the number of muscle fibers. Similarly, hypertrophy of muscle in response to exercise is due mainly to an increase in muscle fiber size rather than the increase in number. Histology of muscle fibers. Muscle contraction is much easier to understand when we consider the structure of a muscle fiber. 9.3. The plasma membrane of a muscle fiber is called the sarcolemma. Two delicate connective tissue layers are located just outside the sarcolemma. The deeper and thinner of the two is the external lamina. It consists mostly of reticular collagen fibers and is so thin that it cannot be distinguished from the sarcolemma when viewed under a light microscope. The second layer also consists mostly of reticular fibers, but it is a much thicker layer called the endomysium. Along the surface of the sarcolemma are many tube-like innervations of the sarcolemma called transverse tubules or T-tubules. They occur at regular intervals along the muscle fiber and extend inward, connecting the extracellular environment with the interior of the muscle fiber. 9.3b. The T-tubules are also associated with the highly organized, smooth endoplasmic reticulum called the sarcoplasmic reticulum. In skeletal muscle fiber, other organelles such as numerous mitochondria, energy-storing glycogen granules, are packed into the cell and constitute the cytoplasm, which in the muscle cell is called the sarcoplasm. The sarcoplasm also contains numerous myofibrils, which are bundled of protein filaments. Each myofibril is a thread-like structure, approximately 1 to 3 micrometers in diameter, that extends from one end of the muscle fiber to the other. A myofibril contains two kinds of protein filaments called myofilaments. 9.3c. Actin myofilament or thin myofilaments are approximately 8 nanometers in diameter and 1,000 nanometers in length, whereas myosin myofilaments are thick myofilaments are approximately 12 nanometers in diameter and 1,800 nanometers in length. The actin and myosin filaments form highly ordered units called sarcomeres, which are joined end to end to form myofibrils. 9.4a. Actin and myosin myofilaments. Each actin myofilament is composed of two strands of fibrous actin, F-actin, a series of tropomyosin molecules, and a series of troponin molecules. The two strands of F-actin are coiled to form a double helix, which extends the length of the actin myofilament. Each F-actin strand is a polymer of approximately 200 small globular lumens called globular actin G actin monomers. Each G-actin monomer has an active site to which myosin molecules can bind during muscle contraction. tropomyosin is an elongated protein that winds along the groove of the F-actin double helix. Each tropomyosin molecule is sufficiently long to cover 7 G-actin active sites. Troponin is composed of 3 subunits, one that binds to actin, a second that binds to tropomyosin, and a third that binding site for calcium. The troponin molecules are spaced between the ends of the tropomyosin molecule and the groove between the F-actin strands. The complex of tropomyosin and troponin regulate the interaction between active sites on G-actin and myosin. Myosin filaments are composed of many elongated myosin molecules shaped like golf club heads. 9.4b and c. Each myosin molecule consists of 2 myosin heavy chains wound together to form a rod portion lying parallel to the myosin filament and 2 myosin heads that extend laterally. 9.4b, see figure 9.3d. 4 light myosin chains are attached to the heads. Excuse me. Excuse me again. Attached to the heads of each myosin molecule. Each myosin myofilament consists of about 300 myosin molecules arranged so that about 150 of them have their heads projected towards each end. The centers of the myosin myofilaments consist of only the rod portions of the myosin molecule. The myosin heads have 3 important properties. 1. The heads can bind to active sites on the actin molecules to form cross bridges. 2. The heads are attached to the rod portion by a hinge region that can bend and straighten during contraction. And 3. The heads are ATPase enzymes which break down adenosine triphosphate ATP, releasing energy. Part of the energy is used to bend the hinge region of the myosin molecule during contraction. Sarcomeres. The sarcomere is the basic structural and functional unit of skeletal muscle. Because of the smallest portion of skeletal muscle capable of contracting, each sarcomere extends from one Z-disc to an adjacent Z-disc, figures 9.4 and 5. A Z-disc is a filamentous network of protein forming a disc-like structure for the attachment of actin myofilaments. The arrangement of the actin myofilaments and the myosin myofilaments give the myofibril a banded or striated appearance when viewed longitudinally. Each isotropic band or I-band includes a Z-disc and extends from each side of the Z-disc to the ends of the myosin myofilaments. When seen in longitudinal and cross sections, the I-band on each side of the Z-disc consists only of actin myofilaments. Each anisotropic band or A-band extends the length of the myosin myofilaments within a sarcomere. The actin and myosin myofilaments overlap for some distances at both the ends of the A-band. In a cross-section of the A-band where actin and myosin myofilaments overlap, each myosin myofilament is visibly surrounded by 6 actin myofilaments. In the center of each A-band is a smaller band called the H-zone, where the actin and myosin myofilaments do not overlap and only myosin myofilaments are present. A dark line called the M line is in the middle of the H-zone and consists of a delicate filament that attaches to the center of the myosin myofilaments. The M line helps hold the myosin myofilaments in place, similar to the way the Z-disc holds actin myofilaments in place. Figure 9.5, B and C. The numerous myofibrils are orientated within each muscular fiber so that A-bands and I-bands are parallel. Myofibrils are aligned and thus produce a striated pattern when seen through a microscope. In addition to actin and myosin, there are other less visible proteins within sarcomeres. These proteins help hold actin and myosin in place, and one of them accounts for muscles' ability to stretch extensibility and to recoil elasticity. Titin is one of the largest known proteins, consisting of a single chain of nearly 2,700 amino acids. It attaches to the Z-disc and extends along myosin myofilaments to the M line. The myosin myofilaments are attached to the titin molecules, which help hold them in position. Part of the titin molecule is the I-band functions as a spring, allowing the sarcomere to stretch and recoil. The primary function of skeletal muscle cells is to generate force by contracting or shortening. A parallel arrangement of myofilaments and sarcomere allows them to interact, which causes muscle contraction. The interaction is described by the sliding filament model. figure 9.6. Sliding filament model. It is the shortening of sarcomeres that is responsible for the contraction of skeletal muscle. In a sarcomere, the actin and myosin myofilaments slide past one another but remain the same length as when the muscle is at rest. When the myofilaments slide past each other and the sarcomeres shorten, the myofibrils also shorten because the myofibrils consist of sarcomeres joined end to end. The myofibrils extend the length of the muscle fibers, and when they shorten, the muscle fibers shorten. Groups of muscle fibers make up a muscle fascicle, and several muscle fascicles make up a whole muscle. Therefore, when sarcomeres shorten, myofibril muscle fibers, muscle fascicles, and muscles shorten produce muscle contraction. During muscle relaxation, the sarcomeres lengthen. For this to happen, an external force must be applied to a muscle by other muscles or by gravity. For example, muscle contraction causes the joint, such as the elbow or knee, to flex. Extension of the joint and lengthening of the muscle result from the contraction of muscles that produce the opposite movement. See Chapter 10. However, knowing the mechanism by which muscles shorten is a small step towards understanding muscle contraction. What stimulates the sarcomeres to shorten in the first place? In order to fully understand muscle contraction, we must first consider the electrical properties of skeletal muscle fibers. Muscle fibers, like other cells in the body, are electrically excitable. A description of the electrical properties of skeletal muscle fibers is presented next, and later sections illustrate the role of these properties in contraction. Axons and neurons extend from the brain and spinal cord to skeletal muscle fibers. The nervous system controls the contraction of skeletal muscle through these axons. Electrical signals called action potentials travel from the brain to spinal cord along the axons to muscle fibers and cause them to contract. Ion channels. In order to understand electrical properties of skeletal muscle fibers, a review of the permeability characteristics of cell membranes and the role membrane transport proteins play in the permeability will be helpful. The phospholipid bilayer interior is a hydrophobic environment which inhibits the movement of charged particles, particularly ions, across the membrane. However, the basis of the electrical properties of skeletal muscle cells is the movement of ions across the membrane. Recall from Chapter 3 that ions can move across a membrane through ion channels. There are two major types of ion channels, non-gated or leaked and gated. These two channel types contribute to the electrical properties of both resting cell and stimulated cell. In resting cells, the non-gated ion channels allow for the slow leak of ions down their concentration gradient. Like all membrane transport proteins, leak channels are specific for a particular ion. The gated ion channels are more important in stimulated cells. It is their presence that governs the production of action potentials. There are two major gated ion channels. Ligand-gated ion channels. Ligand-gated ion channels open when a ligand or chemical signal binds to a receptor that is part of the ion channel. See Figure 3.8. For example, the axons of neurons supplying skeletal muscle fibers release ligands called neurotransmitters, which bind to ligand-gated Na+ channels in the membranes of the muscle fibers. As a result, the Na+ channels open, allowing Na to enter the cell. Voltage-gated ion channels. These channels are gated membrane channels that open and close in response to a particular membrane potential. When a neuron or muscle fiber is stimulated, the charge difference changes, and particular change causes certain voltage-gated ion channels to open or close. The voltage-gated ion channels that play major roles in the action potential are voltage-gated Na+, K+, and Ca2+ channels. For example, opening voltage-gated Na+ channels allow Na to cross the plasma membrane, whereas opening voltage-gated K channels allow K+ to cross potassium, and opening Ca2+ calcium channels allow Ca2+ to cross. Differences exist across each plasma membrane. Resting membrane potential. Electrically excitable cells, like most cells, are polarized. That is, the inside of the most plasma membranes is negatively charged compared to the outside. Thus, a voltage difference or electrical charge difference exists across each plasma membrane. This charge difference across the plasma membrane of an unstimulated cell is called a resting membrane potential. Figure 9.7, see Figure 11.14. Action potentials cannot be produced without a resting membrane potential. The resting membrane potential is the result of three factors. 1. The concentration of K plus potassium inside the plasma membrane is higher than that outside the plasma membrane. 2. The concentration